It's like, I'll just stay here.
Almost looks like a body outline or something like that.
Yeah, these waves
Crime
are
scene?
Yeah.
interesting. The one in current's still fine, though.
And there's an anemone
Yeah,
on that rock, too.
Anemone. So another question is, have we ever come across carcasses on the seafloor? And I do believe we have on previous expeditions. And if you go look at our gallery and different images and videos there, you'll see different carcasses that have been captured on these expeditions. Thank you for asking.
So Dave, can we pop up the chart? I'm going to ask Emma to tell us where we are in this whole progress of this. Great, thank you. Emma, do you want to tell us our progress, where we are in this whole?
Sure, yeah. So we are in the world about 100, 115 nautical miles southwest of Johnston Atoll. And we've just been hanging out around Johnston Atoll this entire time, looking at all the cool sea mountains and stuff. And this is the current seamount we're on. And if you look out wide, all of these more spaced outlines are about 100 meters of depth change. So it goes from dark blue deep to this kind of yellowish shallow and at 100-meter increments. But we have loaded in some more precise lines closer to where our waypoints are and where we're traveling, just so that we can kind of assess where we're going with more accuracy. And these are 30-meter different, 10-meter different changes in height between the blue to the yellow. And we have 9 or 10 waypoints, and we are starting down here yesterday at about 2,500 meters of depth, and we have been slowly climbing up this ridge, and we are in between waypoints 7 and 8 right here. And you can see that there is a black line that connects all the waypoints, and also a red line, and these are the trails of where the ship, Atalanta and Hercules, have been. And zooming in here, you kind of see a mess of color because we were spending a lot of time in one area collecting that sample. But the red is Hercules, and that's where Hercules has been going around. So you can see Josh going back and forth looking at all of the cool corals and rocks around. You can see Atalanta, which is higher up in the water column, and that's in the blue. And it has been staying in just about one spot since we've been stopped. And the black is the boat. So yeah. And Atalanta has this big circle around it, which is 30 meters. And that's how long the tether from Atalanta to Hercules is. So generally, you're staying in that circle. And there are 2 dots for Hercules. One of them, there was 2 different ways of telling where Hercules is. And so right now, they're a little out of sync, so I'll fix that. But they're better. One is better than the other, depending on where we are on the seafloor and stuff. So yeah.
Can you zoom out a little bit? I want to show the hard left turn we made.
Oh yeah.
Yeah, and for people who are tracking along, right where you see the hard left turn, that's where there's a beautiful garden of corals that we saw in a relatively nose or flat portion of this climb. And we actually saw some nice stuff on the a little farther to the west along the sharp cliff there. Some nice
yeah,
corals
this is
there
where that big cliff was, all the way from here to about here.
Yep. Okay.
So half a nautical mile.
Great, thank you.
Yeah, or a quarter nautical mile.
Again, very spotty across this plain.
Yeah.
Lots of crinoids.
I'm expecting to see more of the cucumbers and the urchins.
Yeah.
This is what they like. And we already have seen a few.
These rocks have a lot of manganese incrustation on them with the mossy and botryoidal texture.
Are you expecting it to still be limestone underneath?
I'm pretty sure that's what it is. I mean, I did get that rock just to be sure.
What's that a rock?
Yeah, it looks
Yeah.
like it, yeah.
The rocks.
You've been really good at spotting rocks today.
Thank you.
Yeah, it's a talent. Are you surprised that we haven't really seen these sea cucumbers?
I mean, we've seen a couple up here.
Really?
Uh,
I thought just the one purple one.
I think I saw a lighter colored one that we just sort of zoomed by.
Yeah.
Oh, cool.
So, I mean, it's like I'm not expecting them to be seen every 5 minutes. You know, they're not going to be that dense, but this kind of tracks with what I would think.
Mm-hmm.
It's interesting that we still see so many crinoids up here since they're suspension feeders.
Yeah, we haven't seen any of those nodule rivers either.
No,
in this
that's
area.
true.
We're starting to see some ripples here, though.
Yeah.
Yeah, maybe we will see one.
But I just wonder, I mean, if this is limestone, I mean, the other ones we saw were primarily basalt kind of in the core and then coated. So I just wonder if we'd get that sort of feature here.
Yeah, it's really just a lot of those yellow crinoids seem to be dominating.
There always seems to be little crinoids on these larger outcrops.
It's another good patch of that detritus.
Yeah.
There.
So Liz, what do you think is the limitation on the number of creatures in this area? I'm just, because it seems like there's a lot of good places where you could live, but they all seem to collect in one sort of area.
Yeah. I think they're trying to get higher up into the water, especially because these ones aren't stalked. For the crinoids specifically, like this rock, you can see it's kind of sticking up in comparison to everything else. So that's what I would think.
So you think it's just, there's that much more of an advantage feeding-wise, or?
Enough that that's where they congregate.
Yeah.
Yeah.
Because I would think like on land, you'd have almost anything that's higher up would have something on it.
Yeah.
Yep.
Yeah, it's very patchy. And I'm not sure, like there has to be something driving this patchiness, but it's hard to like look at a rock and be like, well, why is this the rock that everyone wants to be on?
Is that another rock or shadow or an enemy?
I think that's a shadow. The botryoidal texture makes the shadows look like animals.
Yeah, it really does.
No, I mean, we're at waypoint 8 with 10 to go. I mean, we estimated about a 20-hour. And so we, deployed yesterday about 14 hours ago?
I think we were in the water at like 8:30.
Yeah.
So, okay. So like theoretically back up at 4:30.
Gonna miss dinner. No, I'm just kidding.
No.
Yeah, lots
Another
of
deceptive
crinoids.
shadow.
Is it though?
The bright yellow is really cool though.
Yeah.
Ferns.
Oh, and
Next to the stalk, there might be
sponge.
something.
Oh, yeah.
Another palm tree.
It's a truffula tree.
Were you looking at that thing?
Yeah, but yeah, it looks like it might be a shadow.
Yeah, I
Zoom in,
think it's
please.
a weird shadow. Oh,
Is
they're gonna leave again.
Is that sponge alive, you think, or?
I think it is.
Yeah.
It might be. Oh, that one's missing an arm.
Come back, friend.
You're like,
That's okay, they're unionized.
Yeah.
Come
Yeah.
wide, please.
I mean, I wonder how long it took them to climb up.
Yeah.
All the hard work.
Okay, we got a question coming in the chat from Ben Thickboy. He wants to know, how big do chaunacops get? According to Google, what I found was about roughly 184 millimeters is the average. I'm tired.
I was like, dang, what source is that?
Right, that's Wikipedia.
yeah, see? So we don't use that. No.
Right, so the average length's about 110 millimeters,
Okay.
but they do reach 184 millimeters in length. But would you have any other ideas, Liz, there?
No,
Is that good?
I believe
Okay.
that.
All right.
They're not
Thank
very
you.
big.
And are there any arachnids that live in the ocean? Isn't there one that's related to,
I
they're
don't
in the arachnid family? I thought it was like the horseshoe crab or something.
Oh.
Look at the
There's
weird
one that was
detritus pile
in
to the
that.
right.
But good question,
Yeah, it's
I
a
don't
big
know.
one. Yeah, this is really crinoid land.
Come
Yeah,
wide,
thanks. Yeah, because sea spiders aren't actually arachnids at all.
Oh, they
No, they're
aren't?
arthropods belonging to the Pycnogodia.
Oh, yeah, the horseshoe crabs are spider relatives.
Oh, cool.
Oh, yeah, it does say, which means they are ocean-dwelling animals related to crabs and spiders, apparently, according to Monterey Bay Aquarium.
Oh, good, I'm glad
But
you're
are they
looking at
actually in the group arachnid?
No, I don't.
Oh, they're arthropods.
Yeah, they're arthropods.
Yeah.
Oh, they come in the arthropods.
Yeah.
Thank
Tumble down
you.
Sponge there.
That one laying
Oh,
down?
is that the first? Oh, it's not the same type of sponge though. It's not a ferried sponge.
Look at your
No.
glass.
What?
I
Looks like
still
a
don't
glass
know what the
sponge.
cheese sponge is. That.
Did you say cheese sponge?
Yeah.
Really?
I don't think that's what this one is.
No. What is it?
I don't know.
I don't know what she was referring to.
I think it's the really big yellow ones.
Oh.
Yeah, I think this is the mating season for horseshoe crabs right now too, June.
Oh, yeah? Do you see them out in Rhode Island?
Oh yeah, the salt ponds, there's a lot of them. And
Cool.
they're very characteristic tracks, and the female is larger and the male is a little smaller. You kind of see them paired up going around the pond.
Are they specific to the Atlantic, or do we find them over here on the Pacific
Yeah,
too?
I don't think we have them in the Pacific.
Right?
Oh, is that a sea pen?
Yeah, looks
yeah.
like it.
Is it alive?
Zoom in, please.
Oh.
Oh, yeah.
Wow.
So darker in color than the sea pens
Yeah.
we've seen up until now.
It
Yeah,
is.
it's another one next to the right. Or is that a
Oh, yeah.
tuber?
So, I feel like I recognize the species, but I can't remember its name.
So are those tiny little nodules collecting, or just little rock shards?
I think they're just little shards. Could be
Come on,
biology.
Thanks,
I wonder,
Steve.
too. Yeah, they could be a little.
I think the current has died down a bit.
Yeah, it's still there though. It has died down a little bit, yeah, I'd say. Probably sweeping over the top of that.
Mm-hmm.
Right there.
So you think that maybe a Goldilocks effect with the corals? Maybe the current's too strong here.
Oh, maybe.
Yeah, we are seeing lots of ripples on the sides.
This is the move.
Oh. All right, we got a question for our ROV pilots. What are your favorite things to sample? What are your favorite samples to collect and why?
I don't like urchins.
But you did such a great job.
Don't you like a challenge?
Is it rocks? Do you like to sample?
Yeah, I always like sampling rocks. It's always kind of fun.
They don't run away.
Yeah. I don't know, like challenging things like when we're,
Like
I
that
think
sponge.
trying to come up with like creative ways to sample, because the goal is not to hurt the animal or the rock even. Like you're not trying to damage anything, so you want it to be I want it to be as a perfect sample as I can make it. So coming up with ways to pick up delicate things and not damage them is always fun. It's more just about, like, you can get really creative, right? There's not just one way to sample something. So I don't know. I don't think I have one in particular, but for me it's just kind of like trying to come up with creative ways to make it a nice clean sample so it arrives on deck in good condition.
Thank you, Josh. This looks like crinoid.
I liked your determination on that sponge.
Yeah.
In the crevice.
Yeah, those are tough. Like when you
That
got
was
to
hard.
wedge the ROV into a position to get at something,
Yeah.
it's kind of fun. It's really all about the setup. It really starts with giving you the right angles and like kind of knowing how you're gonna go about sampling something. So getting the ROV set up in the right spot is critical. Can we zoom in, please?
Oh, I missed that.
Oh.
Good spot. It's a little tiny synallactid.
Do you have that one already? Is this a new species for
I
you?
actually think it's going to be a little small for me.
Oh, not enough.
Not going to get a lot out of its guts.
Little baby tiny holothuirian.
Yeah, you're good in it.
Oh, come on, please.
'Cause that one's looked like it was less than 10 centimeters.
Oh yeah, way less, yeah.
So.
I can try and get a... actually, you know what? Maybe. This close. A little less than 10, I would say, yeah.
Yeah. Oh, here's maybe that, like, what we were calling a riverbed.
Yeah, I'm almost kind of tempted if we see something that has more density to get a scoop to compare and contrast the nodule fields.
Oh, there's another umbellula on the bottom, I think.
Is that an urchin there?
Looks like it.
Small one?
Yeah.
Much smaller, yeah.
Hungry for another?
No, we're good.
What urchin? I see no urchin.
Yeah, that one's less than 10 centimeters. What's this white thing over here? Might be sand.
I was like the edge of the rock or something. Maybe.
Yeah, sorry. No,
That's all right.
So we're approaching waypoint 8 and kind of going to waypoint 9. Do we want to keep going towards the middle, or do we want to cut a little closer to the ridge again?
Yeah, let's go to the ridge for a bit. I mean, kind of hug that ridge.
Yeah, it seems like this is all we're going to get in the middle.
Yeah, I think we've characterized this region.
Okay.
Crinoid sand,
Sound good?
shadow.
Urchins.
So one big question that's come in here are, is seapen or rock pen also a colonial animal?
Yes, it is. It's actually another type of octocoral.
Sound good to you
Nice. Thank
too?
you.
Oh, like right here,
change.
Right?
Sure.
bridge nav?
Yeah, sea pens
Okay.
are octocorals, yes.
Thank you.
And then
Hey,
another
can we change
curious.
course to
Oop.
220? Oh wait. 200. My bad, thank you.
There it looks like, I can't tell if those are nodules or just rock. That
Yeah.
looks like.
I mean, the sediment cover is just enough. It looks
Yeah.
like a lot of the rocks with botryoidal are nodules.
These rocks are tricky.
I mean, it's essentially the same process, right? Like the reason they have those little bumps on them is it's basically nodules but forming on a surface. Is that a shadow? Yeah. It's a fish-shaped shadow. Whoa.
Ooh.
Yeah, there's a,
Crinoid, yeah.
or is that a resingid?
No, I think it was a crinoid.
Okay.
He's gone to the trough or something.
What? What? We're rocking and rolling all around.
Ooh.
Did change his heading. It's like now he's changing it again. He's like, nah, that's not working.
I love being at the highest point and rolling around.
Another rock that's just like a hot spot for crinoids.
I know, and they're all the bright yellow ones too.
Yeah.
You know what they say, location, location, location.
Prime real estate.
We should be coming up on the ridge soon, or the drop-off.
Yeah, I don't know what that was.
I think that was a crinoid with a weird shadow.
Yeah. Driving along an old, dried up underwater seabed, or riverbed here.
Dried up?
It is funny how things can look dry down here.
Yeah.
This is a seapen, I think.
Yeah, that's what
Another
it's like.
one?
Yeah, I was just wondering why there aren't more of them down here.
Yeah.
We'll zoom in, please.
Is that a brittle star friend?
Yeah.
Oh.
Or, no,
What?
that's just the tip is all droopy.
Aw,
Oh dear.
poor seapen.
Did it just droop over on its own, or is that the current pushing it over? It looks like, oh, and then it came, it's getting lifted up again, yeah.
Oh, here's a coral.
Oh,
Zoom in.
Metallagorgia.
wow.
It's leaning over a lot.
Oh, stalk like that?
Yeah.
What's going on here?
Yeah, I think it's just got a stalk that's like really bent.
Are those normally on
But
stalks?
like,
Yeah.
yeah?
That looks like a fake start.
Wow.
Looks like a wire.
Yo, come on, please.
Definitely a wire, as in, why are you here?
More crinoids.
Yeah, a lot of them.
Yeah.
Another seapen.
Yeah. And then another stalked sponge.
And you can tell by the asymmetry of these ripples that it's probably flowing to the left here,
Nice.
the current. Another seapen.
Let's zoom on that. It's hard to tell. It looks like it might be something dead or maybe a tube from a tube
Tube worm.
worm.
Zoom in, please.
Oh. Yeah, I guess that's a seapen.
Yo, come light.
And you know we're going to get to the edge where all the corals are just as we do a shift change.
Of course.
I think we had quite a bit of excitement this time.
We really did.
Yeah, we
That
did.
was a good one for sure.
That cliff was awesome. That urchin, awesome.
Yeah.
Peek over here and see if we can see.
It's a big stalked one over here.
Oh yeah. Firework.
Yep, here's you.
Atalanta's teleporting again.
Huh? Oh.
We'll zoom in, please.
Definitely current there.
Okay, we'll come wide.
That little red thing hanging on there, did you see that?
No.
Off to the left?
Uh-uh.
A little bleb of something.
Huh. Yeah, there's not much here.
Yeah, I haven't quite gotten to that ridge yet.
Messages? Okay.
All right, I got a couple more questions coming in. Do the ROVs have an onboard power
Yep,
and
that's the edge.
autopilot?
The power all comes from the ship.
Okay.
We do have autopilot. It's what we call, refer to as auto XY, but it's a closed loop control. So we can go into full auto mode and have the,
Good tugging.
yeah, I'm trying to get it back over. You can kind of push buttons and fly the ROV as well. So ask it to do specific headings and distance off bottom at a certain speed, a certain distance.
Mm.
But
If
often
they-
when we're exploring, we just kind of free fly.
Okay, thank you. If they get disconnected from the surface ship for some reason, would you have to send another vessel down to retrieve them?
More than likely, yeah.
Okay. And how large do sea pens get?
I don't know the upper limit, but I've seen some that were like maybe 1/2 meter tall.
Whoa,
Yeah, they can get pretty
that's
big.
really tall.
Okay.
Yeah, that's a big one there.
And how much longer is this dive going to last? I think we're going to be going for a while longer. We've got, what, 2 more waypoints to get to?
9 and 10.
Yeah, we initially scheduled it for 20 hours. We're about 16 or 14 in,
Okay,
so.
definitely over halfway.
Can we look at this?
But the 20 isn't necessarily hard and fast.
So feel free to take a nap if you need to and come back and join us when you're done.
Okay,
There's
thanks.
a watcher who
Zoom in,
wants
please.
to, yes.
You, Dave.
There's a viewer that wants to get some sleep, but doesn't want to miss the show.
Dave wants to nap.
That, I want to
Mushroom.
say umbellula, but
Yeah.
the dark color is, yeah, I think that's what it is. It's a seapen.
Pretty.
Cool.
But I'm not used to seeing them dark. Usually they're all white, so I'm not confident.
This thing looks crazy.
Oh, come my please.
Oh, okay, yeah, they can be dark in color.
Yeah, what's our viewership like now?
That looks like another, oh my god, what are they called? Brizingid.
Bazinga.
No, that was a while ago.
Aloha, Germany. Mahalo for sharing that you're, wow, it's near midnight now. Yep, get some rest, come join us. We'll be having a lot more dives and expedition, or on this expedition, so. So a few of these questions we've answered before, but I don't think that we've put any bait on the ROV to attract or examine fauna. Josh?
Yeah, not very often. I don't
Is that
think we've
something that
actually
you
ever
guys
done that
do?
with Hercules before. But yeah, it's certainly could do that. But yeah, I don't think we've done that.
And I think we were talking about it, Liz, yesterday. What was the strangest thing that we've seen at the bottom? There was something that we saw. It was that that marking, that scarring on the rock from an anemone that we couldn't figure
Oh,
it
yeah,
out, and it was
odd.
it was pretty odd. Anything else? How fast can Hercules go if you wanted to chase a fish? Hey.
Her can go a couple of knots when it's for short periods.
Somebody wants to know if you're going to upload the urchin dunk as a clip to your YouTube channel.
Oh, that's a highlight reel for sure.
I think that was is epic. Yep, that was a good one.
Yeah. Thank you.
Yeah.
Okay, we'll come wide.
What does that mean? I just slowed it down because I just
Yeah, I agree.
and I didn't know
think that's a palopatides.
Are we starting to get a little more slope now? A little steeper?
Closer?
Yeah. That little ledge is over there somewhere.
Mm-hmm.
There's a dead coral skeleton over there.
All right.
Yeah.
Yes, the comm lady went slightly quiet on the mic for this cruise just to open the floor to our scientists and other communicators to share experience down here. But it's been quite fun to chit-chat and answer your questions. So keep them coming. We've got a few more minutes before the You're Muted watch transitions into the sea pickles.
What was the name of the other watch again?
Oh, good griefy. Something Dan
Delta
and
Dan.
Delta Dan and the benthic
Storm.
storm.
Yeah.
Delta Dan and the benthic storm.
Look at that.
They'll play music for you when they come in on their watch. Just kidding.
Just kidding.
Yeah, it looks like we may be finally coming to the edge of the scarp too.
Ah.
Sediments are dropping off.
So some pretty awesome creatures that we've seen today. We've seen a lot of corals,
Zoom in, please.
sponges, crinoids have been pretty present throughout this dive. We've been able to capture some sea cucumbers, which has been really nice.
Is that a tube
Look
dwelling
at-
anemone?
And there's a tube growing anemone right there.
Yeah, it's cool.
Yeah, very cool.
Pretty neat.
Yeah, we've been seeing a lot of anemones. Do you want to, so that was a question here. Oh, they just put it on YouTube. Hi all, what creatures have you seen so far today? So I listed a few. Liz, if you guys want to touch on some others that might have been.
I know that
Caught.
they saw a Dumbo octopus
come
in
on,
the
please.
shift before
That's
us.
right.
It's
And
pretty
then
cool.
Chaunacops. And it looks like the Duncan urchin just hit YouTube.
Really? Already?
Yeah,
That's
Wow.
awesome.
it's trending.
Yeah, you're
It
blowing
was
up,
just
Josh.
that good.
Is it really? It's not trending
Yeah.
already, is it?
No.
I
You're
have
just
no
joking.
idea.
Oh, okay.
Somebody
I was
sent
like,
us the link.
From 0 to something, it's trending.
Is there the audio on there? Us
I
cheering
hope
at
not.
the
I
end?
hope not. Yeah,
Me
I was
too.
screaming a lot.
Yeah.
Oh, I'm sure there was.
Things
Couldn't
got
help
pretty
myself. Yeah.
Hey, but at least nobody said bad words.
I know. Josh's poor ears.
Sorry, Josh.
Yeah.
He's
That's
like,
the best
everyone,
part.
calm
Just
down.
let's relax.
Everybody calm.
It was too hard to calm down.
Yep, it's on there. It's on the Nautilus Live Channel 1 stream.
Oh my gosh.
Just
Boom,
everyone relax.
shalaka-laka.
I do this all the time.
Is that cool?
Oh, you're muted. Don't leave. Sad face. We'll be back at 8 o'clock tonight to 12. And if we have our blue water show, if we're ascending, we might host our blue water show again.
We should have a different theme every time.
Yeah.
Yeah.
Yes,
The
there's
Blue
been
Water
a lot of
podcast.
suggested
Yeah.
themes in our chat. So,
I think we all need some sleep right now. That's why we're not
Yes.
very exciting.
right, yeah.
No,
we
you-
think we
I'm
pooped
standing
out
up
this
just so I
morning.
don't fall asleep.
All the excitement has been pushed out about less than 45 minutes ago.
Yeah, that urchin really took the last energy out of me.
Uh-huh.
Yeah. I had a lot
I can
of
sleep
adrenaline.
good tonight.
Yeah.
I think, yep, it just took it out. All the coffee I drank did nothing for me.
Yeah, same. I had a Coca-Cola and a coffee.
Oh, boy.
And I'm still sleepy.
Get a Coke coffee, or you had them separately?
A
Whoa,
little bit of
you
a mixture in between because I didn't clean the
Cray
mug.
No. Have you had the coffee Coke?
No, I haven't.
Oh, it's so good.
Yeah?
Does it taste like Coke?
It tastes like coffee.
And it
It
looks
tastes
like coffee?
Coke.
Ew.
Tastes like
Yum.
Coke, looks like
Coffee
coffee?
cola. Yeah, coffee cola. It's so good. My drink of
Neon
choice.
Yellow.
You pepped us up a little bit there, guys. Everybody.
In Rhode Island, they have this drink called coffee milk. It's like a coffee syrup with milk.
Oh, that's weird.
Yeah, what?
They love it.
So it's mostly milk,
Is it
but
cold?
with flavor
Yeah.
of
Wait,
coffee.
what do they have?
Yeah.
Oh my god.
Coffee milk.
Do they
Coffee
serve it
flavored milk.
cold?
Oh
Yeah,
yes, yes,
yeah.
yes, they do.
I feel like that's how my mom drinks her coffee. It's mostly milk
Yeah.
with just a little bit of coffee.
With the splash
Lots
of
of cream.
coffee.
But it's cold. It's almost like a milkshake.
Oh,
Or
They
Rhode
sell it.
interesting.
Yeah, or in Rhode Island, they call milkshakes cabinets, so it's tough to get used to.
Cabinets.
Yeah,
What? That's the most
They
bizarre
call
thing.
it what?
a cabinet.
Where's this at?
A coffee
Rhode
cabinet?
Island.
East Coast, man.
Don't they call like water fountains bubblers or something
A
too?
bubbler.
Isn't there some New York drink where you put like ice cream and sparkling water or something?
Ooh,
I might be
that
making
sounds
that
kind
up.
of gross.
There's something like that.
At first I thought of
Well,
a root
that's-
beer float, and
That's
then
an
I
ice
was
cream
like,
soda. If you do
Always
ice,
goes
ice
back
cream
to
Yummy, I
soda.
love ice cream soda.
Always back.
I love cream soda. Here we go, it's a
In Rhode
soda
Island, we're
kick.
drinking it. In
But
Galway.
New York
Hey,
also
you
has
guys
egg
got
cream.
it. I
Oh,
think
yeah,
we
that's
pepped
right,
up.
egg.
They actually do call them bubblers, though. I didn't believe
Ooh,
it until I
coral.
went there.
is that a bamboo?
Yeah, let's get a zoom on that.
It's tall.
It's very
Oh,
fuzzy.
zoom in, please.
Ooh. Is that a new one or just a different, more
looks like a
branching
bamboo.
on it?
Yeah.
Yeah, I see nodes.
Oh boy. I used to have a pet tarantula. The question is, who wants a pet tarantula? I used to have one. I don't have
I'm more of a
anymore.
marine person. I have a clownfish.
Oh,
He's a black and
that's
white
a
oscillarius.
Nemo. What's his?
Yeah.
What's the clownfish's name?
Bo, because he's Bo. He's black and white. Sorry, and I named him after Chadwick Boseman when he passed.
Oh. Milk with Coke. Wasn't that a Laverne and Shirley thing? Didn't Laverne add milk to her Coke?
Pepsi, though, for Laverne.
Pepsi, thank you. Milk with Coke called a brown cow. Delish. I don't know if Rob's up for jokes today.
That's what it was, a scuttlebutt.
I'm saying jokes inside my head, which is probably
Okay.
better.
That's probably a better place to keep him for now.
A little
Another,
fireworks.
yeah.
Ooh,
Oh, and a
sea
sea star. Nice.
It's a nice iridigorgia.
Yeah, that's what it
We've
is.
seen quite a few sea stars, I feel.
Yeah.
Sponge off in the distance too.
Let me see where that urchin link is.
Yeah, they'll probably be showing the urchin clip at Josh's Hall of Fame induction to the ROV Hall of Fame. Wow, look at this one.
Oh, let's get a zoom on that. Oh, it's a sponge.
Sponge with.
I thought it was a coral.
Wow.
Wow, don't
crinoids.
you disappointed?
Yeah.
Oh, it's a sponge.
Well, I thought it was something we hadn't
Zoom
seen
in, please.
before.
It's another one of those palm tree looking
Yeah.
Yes.
sponges with the...
Oh, look, a brittle star fell off for us already.
That's just packed with brittle stars.
Yeah.
Yeah.
I mean, it
Do you think-
literally looked like a palm. It's the combination of things that make it look like a palm tree. Which one do you think is going to fall first?
It's like a Joshua tree.
There's like a crinoid hanging on by
Oh.
a thread.
The back, there it is.
there
Oh,
he
where'd it go, George?
Yeah, come on, please.
And a shrimp.
Lightning in the crinoid. Sorry. I'm getting bored now. Oh, yeah. Yeah, I don't know.
I'm really looking forward to seeing that anemone we collected.
Yeah.
Yeah.
That was really cool.
Yeah.
I'm a little sad we didn't get to see it swim.
Yeah, I wanted to see it try to run away, but not successfully.
Yeah.
So yeah, it looks like this is somewhere on YouTube, but I can't seem to figure out how to guide viewers to the
I guess
to
what's the
sea
title
urchin
of the,
dunk.
the video?
Copy and paste that up there. Oh yeah.
Where?
No, you can't.
Can't, right?
You could click on the video there. It should link you to YouTube. If you...
Yeah, she's on YouTube. Oh, well then it's in the chat.
Yeah, just put EB Nautilus sample of the year. It should pop up. 2022. What is that? Oh, it's a shadow. Oh, my glasses. Very interesting how quickly it changes.
Quickly what changes?
Like just the landscape, just like so abruptly.
From like rock to sand to cliff to
Yeah.
flat? Yeah.
Okay, so in regards to the urchin clip, we did see somebody did snip it and put it on YouTube, and they entitled it "Catching a Hitchhiker." So we ourselves have not posted anything on our YouTube channel relating to that moment, but others who have been streaming or watching our live streams perhaps have. So that's the best information I can give to you folks right now in regards to that particular highlight.
Well, you can always go back on the live stream too, because of when you watch it, you can actually
Oh, cool.
go
Thank you,
back and
Josh.
search things over again.
Yeah, it happened at, I think the final collection time was 2051 UTC. So
Oh, okay.
if you want to try to go back to around that time.
Okay, so we have a question from someone's daughter who wants to know where in the world you are currently searching the ocean.
Oh,
So we're right now, and
can
I'll
zoom out.
pass that over to Rob. Would you like to share about where we are?
Well, actually, Emma would have a better
Oh,
handle on
Emma.
that.
There you
Oh,
go.
yes. Okay, I can zoom out a little bit
Yeah.
here. So we are at Johnston Atoll, which is nearest to Hawaii. It's like the nearest piece of land.
Mm-hmm.
And if you look at the map, there we go.
So if you look at our live feed channel 3, Emma is showing you our navigation map that we're using to guide us through this area of the ocean. And that
So
gives you a sense of where we're at.
yeah, you can see in this upper right with all the circles, that is the Hawaiian Islands. And they're covered in a whole bunch of text because we've marked them up. But that's
Mm-hmm.
Hawaii. And then you come over to the center left, and we have our circle with all of these places that we've mapped, all the underwater seamounts, so you can see them. And all of the background chart color is parts of the ocean that haven't been mapped yet in this area. So it's pretty cool to see what we have and haven't mapped. And zooming into the middle of the circle, we have Johnston Atoll, which is the closest, I don't know, major piece of land. And to the southwest is where we are. So we are in the Pacific Ocean, kind of near Hawaii, west of it. And we are looking at underwater sea mountains off of Johnston Atoll.
Thank you, Emma. And there's another question that came up that I think you'd be able to address best is how far above the surface do you normally drive?
Do you want to answer that, Josh?
Yeah.
Above,
Sorry, what's the question?
Yeah, I
how
kind
far
of
below the
confused. Maybe,
surface?
so whoever sent that question, if you could rephrase that for us to give us a sense of what you're asking, that would be helpful.
Are they asking how far above the surface of the ocean floor?
Yeah, that's what I think
Maybe,
they were.
that's what I'm
Oh.
thinking, right?
Oh, it's like the altitude of the
Yep.
ROV.
Yeah, maybe.
It kind of varies
Oh,
depending on the terrain. I mean, right now we're reading about 2 meters off the seafloor.
oh, thank you,
Oh,
Josh.
look at that.
Okay, on that note, you're muted is officially off watch. Thank you for sharing the brown cow root beer float soda recipe. And we will see you again at 8 p.m. Pacific time. Ahui ho malamapano.
We're coming up on 9.
Check, radio check.
Video radio check.
Loud and clear.
Thanks.
Hey everybody, how you doing? So the last crew kind of was going along the lip, as you can see, to the east of waypoint 8, seeing a lot of cool stuff, and then decided to come up onto the flat summit to see if there's anything there. And there isn't much, so I'm hoping to go back, kind of due south along the ridge, to continue to look at all the large coral gardens, if that works for you guys.
Okay.
Sounds good.
Due south, Raj. Along the cliff, we like.
Are they seeing a lot of, excuse me, woo. Are they seeing a lot of corals and sponges and down there?
Yeah, apparently, well, let's see, the last watch saw a good amount on the lip, and then these watch that took over after us had huge, really dense coral gardens, so hopefully we get back into that. Here we got all these beautiful yellow crinoids.
Roger that. Other follow-up question. Can you please go over the potential recovery times and or any information with regards to recovery?
Yeah, last I heard, and it wasn't official, it wasn't on the light board of lies, but we're looking to get on deck around 4:30, but I'm going to send out a message to Steve, and we'll confirm that, and I'll get you a pull-up time. We're not going to make it to the top of the gill. I think that's pretty obvious.
Okay. Sounds good.
Raj.
Oops.
Oh, wow.
It's all yellow crinoids all the time.
Yeah.
Do a partial zoom on this rock here, please.
Can you be different colored crinoid in the same species, or would the color indicate a different species?
You know, I don't know. My guess is that they're a different species, but I also know that color can be a terrible way to tell things apart.
Sure.
Bridge nav, 30 meters, 200.
We've got a nice chrysogorgid coral. I think this one is metallagorgia.
Would you like to zoom on it?
Sure.
All right, go ahead and start your zoom there, please, Tim.
Nice little squat lobster up on its branches. Thank you.
Go wide, please.
See that in bubble cam? I don't know if it's just a shadow of a, like a sea urchin or something. It just looked like it had long spines. Not a sea urchin, maybe something else, but...
Maybe a worm?
No.
Worms don't have spines, Jess.
I mean, maybe it was just a crinoid, but in the sand.
Oh, Raj. Our victorgorgia here.
That's a victorgorgia.
So are these crinoids related, or they just happen to all be in the same area?
We would have to sample them to find out. But there are genetic ways we could tell if they were all siblings or at least distantly related.
Cool.
My guess is that there might be a few that are distantly related, but they're all just in a really good spot for whatever they're feeding on.
Hey, mind if we do introductions so I can check audio levels?
Tim, your audio levels are really low again.
Correct.
First check down.
Should we do introductions and get all our audio levels balanced?
That's a great idea.
That's what he just said.
Great stuff,
Oh,
Jess.
really? Yeah. I'm sorry.
It was so
No,
low
no.
that she couldn't hear the suggestion.
Exactly, which is the whole
It's a
purpose.
great suggestion.
Yeah.
I did not hear it at all. I'm so sorry.
All right, so I'm Onye, your Science Communication Fellow aboard the EV Nautilus. We are still exploring our 6th dive site around the unnamed seamount, number 13, near Johnston Atoll. Apparently, we've seen a variety of deep sea marine life, such as a Dumbo octopus. I can't believe we missed that. Several coral gardens, and
Go ahead and push in and move some screen, please.
how do we pronounce this?
Tromikosoma.
Chaunacops fish,
The
yes.
cutest fish in the West.
Very cute. So join us as we continue to explore this deep sea environment. And yeah, send in your questions and I'll pass it along to the team.
Hi there, I'm Kevin Conrad. I'm a
Hi,
scientist in the back row, geologist out of the University of Nevada, Las Vegas. Happy to be here, looking forward to seeing some more cliff faces and answering any questions on geology.
I'm Meredith Everett. I'm a geneticist at NOAA's Northwest Fisheries Science Center in Seattle, where I work with the Deep Sea Coral Research and Technology Program, studying the biology, taxonomy, connectivity, and environmental DNA around deep sea coral communities. And I'd be happy to answer questions about any of those subjects as we go forward.
Hey everyone, my name is Chanel Vidal and I'm a PhD student from Arizona State University and I'm here as an ocean science intern logging all of the exciting cool corals and sponges that we see. If you have any questions about grad school and what it's like to go from geology to biology for your major, please ask.
Hey, I'm Chris Krasnowski. I'm here as a navigator, also working on the Hercules high-resolution mapping system. Other than that, I am a postdoctoral researcher at the University of Rhode Island.
Hey folks, sitting next to Chris, my name is Jess. I'm part of the ROV team, Plan Herc, and yeah.
Raj. I'm Kylie. I'm sitting in between Jess and Tim. I am piloting at Atlanta.
Okay, and over on the Kylie's left, my name's Tim, and I am sitting in the video engineering seat, which means I am in charge of monitoring audio levels, among other things, as well as video feeds and recordings of all of the interesting, exciting, brand new places that we explore.
It's hard to tell because of sediment. That almost looked like a lateral dike, that long, thin ridge that we just swapped over.
And I didn't want to interrupt the introductions, but there was also just an umbellula.
Unbelievable.
Now we're back into some pill basalts.
What is that square thing?
Yeah, it's a very precarious rock, if it is.
It certainly does not fit the mold.
Go ahead and push it in there, please.
One of those puzzle pieces that you cut into glue, right, Kevin?
Yeah, exactly. Or it could be one of those chocolate mints on top of the pillows that the hotel staff left.
It looks like a rock, right?
Yeah, it might be a very thin limestone piece that somehow made its way over here.
Yeah, interesting.
It's too crusted to be human to be.
Can we get a zoom on the coral that's right behind it too?
Sure, go ahead and come mine, please. Ah, yes, I see. Get myself a bit stable here. Go ahead and push on in there, please. Kylie, you want to turn on the porch light there too?
Sure.
Squat lobster.
Ooh.
Trying to decide if this is the coral we were looking for earlier, but I don't think it's quite the morphology. We had more tentacles down the stalk before.
Looking for a juvenile metallogorgia?
Yeah, one that's kind of a halfway between a metallogorgia and a chrysogorgia. I think this one is just a chrysogorgia.
Gotcha. I'll turn porch back off.
Roger, thank you.
No problem. It takes two to make a thing go right.
Takes two to make it go out of sight. Maybe not go, and adding words here to the song or something.
I like your version better.
So sorry. So we're not copyright infringing, of course.
All right, you're gonna, make it your own.
It's like a large plunger.
What?
The white coral stalk.
Got it. I see it now.
And there's actually a live bamboo coral just to the right of that. I think that's a bamboo stalk. And then, yeah, we've got right under the lasers.
Go ahead and push on in there, please.
Clearly the sea stars have not yet made it to the top of the guyote.
A little wide, please. Get us a better shot of that. Sorry. All right, go ahead and push on in there quick. Come wide, please.
Thank you.
Yeah, sorry about that. That was pretty sloppy.
Ooh, I'm gonna go wide on Atalanta. I just pushed it back so you guys know.
Roger.
Check.
Thank you. Is anybody else in here quite warm right now? Does it feel warmer in here?
It does seem warmer in here.
I'm also warm.
It is a little warmer.
That's because
74.
the thermostat over here is off.
Roger.
All right, it is now back on.
Thank you.
I was recently down in the windshield with this fleece on, so I am all out of sorts.
Sponge?
I think this is a sponge. I think it's just covered in those yellow crinoids.
Rog.
Oh, pretty. Is that, there's nothing like, or what kind of relationship is that?
I don't think this is a official biological relationship other than just the crinoids using and the brittle stars there using this holothuirian to get up in the water column to feed better.
And it doesn't, do the sponges also need nutrients from the water column?
They do. They'll be actively pumping in material through those holes though.
So having this many things on it doesn't like get in the way of its like flow system?
It might, and this sponge actually, if you look at the tissue underneath it, does not actually look that healthy.
Yeah, totally.
See all that fuzz on it? That is not the signs of a healthy, happy sponge, which may be
Bridge nav 3020.
related, though not necessarily causally, to why there are crinoids all over it.
Sure, okay.
It's a very colorful sponge, though.
Yeah. And brittle stars eat the things in the water column also?
Yeah, most of them are deposit feeders or filter feeders.
Okay, so the difference with what they eat between those and the sea stars is that sea stars will eat the polyps of a coral.
Yeah, I mean, there are sea stars that are deposit feeders as well, but a lot of the sea stars we've been seeing are predators.
Okay. And basket stars, what direction do those fall?
I think they're generally filter feeders.
Okay, roger.
Do squat lobsters ever fight brittle stars?
I'm doing a DVR research.
Space?
I could see it happening. I don't think I've ever seen it, but
I saw a shrimp fight a basket star once.
Ooh,
that sounds awesome.
I got stressed out. I'm not sure I remember. I was so scared for him. But that was on the last trip. I'm sure it's a highlight. If not that event, the coral it was on itself, like it was like the size of Herc, and with like so many things on it, it was kind of unreal.
So I just did some looking because I was having a vague memory. There are basket stars that are active predators that have hooks on their really complex arms and can even catch like fish.
Ooh.
Wow.
Yikes.
Interesting.
Note to self.
Yeah.
Where do those live?
Oh, kind of all over. You'll even get, I mean, we've seen one, I think 2 dives ago. There was one up in a coral. And some of them, so you'll get them all over in deep habitats. We see them off California a lot. And then there's shallow water ones that you'll find on coral reefs, like in the Florida Keys.
Do we want to increase the speed so we see more ground back row?
Yeah, unless you want to try to head more southeastwards and dip off the lip. If there is a lip there. We haven't seen it yet.
Yeah, we can.
Yeah, because looking at the contours, I think it's going to look like this for a long time.
Okay, so find the edge.
Yeah, let's try to find the edge.
Okay.
Living on the edge.
Of glory.
Let's do 150.
150, Rog.
Roger.
Bridge nav, hold position, then 30 meters, 150.
And Kevin, any confirmation from Steve?
Negative.
Raj.
Keep us posted.
Yeah.
Yeah, Kevin, so if they want to be on deck by like 4 or so, will be about 2 hours for coming up and for recovery duck work.
Sounds good. Yeah, so until I hear otherwise, maybe we'll start pulling up about 2:30.
Great. Those look like little fingerprints.
Oh yeah. Big fingerprints.
Definitely some sign of current moving along the top here.
I'm gonna do a gauge check.
Sure thing.
Excuse me.
So when will we know this if this is officially a seamount versus a guyot?
Oh, I'm 100% confident it's a geo, based on the bathymetry. Rob grabbed a few samples he thought might have been limestone, so maybe we'll check those. I still think we're in the volcanic section.
So how is a geo formed? Sorry.
Geo is formed from wave erosion, so as the island subsides, it kind of cuts down the island into a flat top as it goes under the water due to the waves and the subsidence. And since all these seamounts would have formed at the low latitudes and stayed in relatively low latitudes, they would have probably had coral reefs at some points that would have lasted until they got drowned out.
So what you're saying is a guyot is an atoll that had slacker corals that couldn't keep up.
Yeah, exactly. They all failed Johnstons.
I feel like there's a young adult novel in there somewhere.
Oh.
Onye, what questions do people have for us?
Yeah, we have a question in the chat about do we get any unexpected creatures kind of stowed away in the containers or bins when we dive?
Yeah. One time there was an unexpected octopus.
Oh.
Just crawled in, crawled out.
Is that one of those umbellulas or whatever they're called?
That is an umbellula.
Umbellula, that was it.
Kylie.
And a little fish.
So beautiful. Beautiful.
Good push on in there. That one's
Nailed
interesting.
Right around the stupid side.
I like it.
Gauges and everything are good.
Roger. Away, please.
Umbellulas are a pain to identify because they're one of the few octocoral species that does not have any sclerites in their tissue.
What is a sclerite?
Sclerites, so if you look at most soft corals, if you look at them under a microscope, they have these little either aragonite or calcium carbonate. They're basically grains of sand.
Yes, okay, that's what we were talking about last night, right?
Yeah.
With the scales.
Yep.
And they can be in different shapes?
They can be different shapes, and those shapes are often diagnostic for the species.
Okay, and the umbellula don't have them?
They don't have any.
Sure, how do you tell?
Yeah, so you just have to kind of look at them morphologically and go, well.
It's close enough, right?
Or do what I do and you look at them genetically.
What's, how much, okay.
Is that one of you guys?
I've never
No,
asked
that's
this before, so I'm not really sure what words to use, but like, how does one DNA sequence? Like, is it a lot of, a couple of steps that like you physically do and then you put it into a computer?
It's several physical steps and then you put it in a robot. So the first thing we do is we use an enzyme and salt buffer mix to break down the tissue and kind of liquefy it. And then we add a variety of chemicals, usually salt-based, to basically precipitate the DNA out of that kind of tissue soup.
Oh.
And then we clean up all of the protein and get rid of it. So all we have left is the DNA. And then put that in a buffer solution that keeps it happy. Then we carry out what's called a polymerase chain reaction, or PCR. And what that does is we pick a gene of interest that will tell us what the species is, hopefully. And we, using heat and temperature cycling and a special enzyme called Taq polymerase, make thousands and thousands and thousands and thousands and thousands of just the gene we want to sequence out of that pool of DNA. And then we clean that up and load that on a sequencer. And the sequencer does some complicated chemistry, and we get a DNA sequence out the other side on the computer.
Oh my gosh.
So the sequencer is the robot, right?
The sequencer is the robot.
Okay.
Okay, I have a question. If I understand DNA, is DNA always a double helix regardless of what it came out of?
Kind of, sort of.
Okay, good enough.
It depends on the animal, and there's some other things that can go on with DNA, but in general, it's double-stranded.
Okay, I was wondering how much of what I have for like a basis of knowledge applies to my question or not. But I guess
Coming down a wee bit.
what I'm wondering is, like, if you get rid of all the proteins and you're manipulating it all chemically in a lot of ways, how do you, like, at what stage is it, and is it ever in jeopardy of becoming alphabet soup instead of DNA?
Yes, so one thing that can happen, and one thing that we're very careful about depending on what we're sequencing and what we're doing with the DNA, is what's very common is for DNA to break down into smaller and smaller fragments. These generally aren't so small that we can't work with them depending on what we're doing. So eDNA actually in the environment tends to break down into small fragments. So this fragment that we sequence for eDNA is only about 350 to 500 base pairs long, versus what I sequence when I extract from tissue can be 1,700 bases long.
Oh.
And if you're going to do genome sequencing, you want really, really long fragments of DNA. And so you actually have to be very gentle, like physically gentle with the sample, because just shaking it too hard can break up the DNA into smaller chunks.
Oh.
But if you're doing a DNA extraction, there's a way you can do it that's actually fairly chemically inert. It doesn't use too many nasty chemicals, just basically alcohol and salt. And if you do that on, say, a banana, which has a ton of DNA in its tissue, you can actually use a little pipette tip and actually pull a strand of DNA that you can wind onto the pipette tip that you can see.
What?
It's a great thing to do for kids' demonstrations.
For adult demonstrations, we got bananas on board, I'm sure.
Strawberries too.
Yeah, let's play some games.
For adult demonstrations.
Thanks for answering that. I had no idea.
This is a very neat formation here.
Yeah.
Yeah, got some little black corals hanging off the lip. Seeing 2 different species so far.
Do you want to go ahead and just push in partial on that geology there? Looks great. So it's like it's broken up down below and then a crusty flow on top. Is that what's
Yeah,
up?
it seems like really thin, really heavily crusted flows.
All right, you can go come wide, please.
Unless it is a limestone feature. It's hard to tell sometimes. We might be in a limestone terrain, but it seems like we're still in a pillow basalt field, best I can tell. Yeah, maybe that, well.
What was that there, Kevin?
Nice.
This is cool.
Yeah, I mean, we might be in some limestone terrain mixed in here. So this geo has kind of 2 flat, like the ridge flattens out where we are now, and then it goes to a proper, like, carbonate platform above that. So there still may be some carbonate because it's such thin little layers.
Nice hemicorallium here.
I really like these flows. So cool. Or this pattern.
Yeah. There's much more of that sedimentary nature where it's really thin and blocky.
Thanks, Kylie. I can also stay a wee bit higher here.
A little fish on the bottom.
Roger.
So what are we thinking about continuing on this ridge or continuing along it or?
Yeah, I think we just continue, probably for the rest of the dive, along this steep ridge.
I'll just kind of follow the contours then.
Yeah, that'd be great, thank you.
So we're going to belay going to the waypoints and just stay this contour the whole time?
Yeah, because it's going to take probably the whole dive just to go over the sandy flat towards waypoint 9, so.
Okay.
Might as well see some cool stuff.
Sure thing.
Bridge nav, 30 meters, 185.
It's another crinoid.
That's an interesting texture there.
Yeah, I think we might be in like a limestone region. We could have had 2 reefs on this geo, one a deeper, one slightly shallower. but not popular with the corals.
Kevin, what can the limestone tell you about dating a seamount?
Interestingly, you can get a minimum age from a limestone. So if a limestone is capping on top of the seamount, you can also make out fossil structures from like foraminifers and similar coccolithophores and other small kind of microorganisms that make shells. So from those, if you can identify specific trace fossils that you know formed in certain age ranges, like say one of them forms between 85 and 90 million years, and you find it in there, you know the seamount has to be older than that because the seamount was in place before that limestone came down. So it can give you some kind of bracketing minimum ages. Although sometimes that can be misleading because they could be way younger than the actual seamount.
Does the ferromanganese crust interfere with looking at trace fossils?
Not really, no, because it's just on the surface. So if you get a big enough piece or a drill core, you can kind of see through it. It'll be fresh on the inside.
Do you use the same radio isotope data, or techniques rather?
No, not on those. Those are usually, it's what's called biostratigraphy, and it's just looking at specific fossil assemblages, when they appear, when they disappear, to try to put it into a time range. You know, we have ideas of when those fossils existed based on areas that have age determinations, and so we can then propagate it into this region. You can look at what are strontium isotopes. So strontium is a daughter of rubidium-87, strontium-87. And the seawater actually has a variation in its strontium isotopic composition through time. It has a distinct curve that correlates to times where there was more volcanism versus times where there was more uplift and erosion. And you can try, if you have a bunch of strontium majors on some of the limestones, you can try to match it to where you are in the curve and see if you can get an age from that. But there's no direct radiometric aging method for limestone. Always this old of a limestone.
Is that because all of the carbon isotopes would have decayed?
Yeah, so
okay.
Carbon-14, you can only go back to about 50,000 years using that method before it all decays away. You can't measure it. And then there's some methods that have to do with uranium isotopes, radioactive uranium and radioactive thorium getting fractionated when they form on the seafloor. But those both kind of go back to an equilibrium after about 500,000 years, so you can't really directly date limestone after that.
Zooming in.
Oh, this is a nice feature.
Yeah, nice little overhang.
Sure. Good old pit 10. Zoom in on the slate.
Yeah, I'm leaning towards limestone. That's kind of typically how it forms. And kind of blocky, squarish flows. Not flows, but layers, sorry. And what's that bottle bush looking guy on the left?
That is a black coral.
190.
Oh, it's got cute little squat lobsters. Yay! So adorable.
I like them. So when squat lobsters are pure white, is that a different species or is that a depth thing?
That's a different species.
Okay.
Nice little overhanging structure here. The nice hemicorallium right at the top.
Oh yeah. No matter where you are in this area, you got crinoid friends.
Is that what you were looking for, Kylie?
I feel like I'm finally getting dialed in on the difference between hemicorallium and paragorgia.
So here's a message in the chat. Given the interconnected nature of the oceans for millions of years via currents, why isn't the distribution of flora and fauna more consistent and homogeneous from sea to sea? So many species and varieties found in only one hemisphere.
So that's a concept called biogeographic boundaries, where even if there isn't a, what we would look at and say, visually looks like a strong, physical wall-like barrier, like a rock formation like this, just simple things like temperature or direction of current can serve as barriers from animals moving around. And so for instance, some of the islands and seamounts just to the south of us, there is a large central Pacific current that crosses between us and them, and that prevents larvae from animals on those seamounts from being transported up here. So on this side of that current, we are in what's called a separate biogeographic province than to the south of it. So things are often less connected than they appear, even over time.
Full wide.
There's also a deep dive you could go into evolutionary niche development as well on that question as part of the answer.
There's another crinoid, looks like. Is that one we need? No.
Yep, we've got a crinoid, 2 different species there. The one on the left may be a species of metallagorgia, but maybe not.
Do we know what gorgia means? With the root?
Not off the top of my head, but I would look.
Bridge nav 30 meters, 190.
Some more crinoids.
Yeah. A little outcrop here.
Biogeography is really interesting. On the east coast of the U.S., there's several different regions where currents, I think, play a big role. For example, north of Cape Cod is the Gulf of Maine. South of Cape Cod is, I forget what that biogeographic area is called, but they're very, very different animals. You get some of the same species, but a lot of them are different too.
Yeah, a really classic example if you want to see terrestrial is to change altitude. So if you can go to an area where you can go up a mountain, you'll see a completely different distribution of particularly plant life the higher and higher you go. As far as I can tell from my brief Wikipedia search, gorgia is from a Latin for whirlpool, which with these species would make sense since they tend to grow in spirals.
Nice. I'm going to start calling them whirlpool holes.
Kevin, so you said the rocks that we're starting to see right now are limestone?
I think so. It's very hard to tell sometimes, but given these kind of smooth, irregular textures, we're not seeing that pillar basalt or even columnar. We're not seeing distinct basaltic kind of structures. We're kind of now seeing a mixture of these large, smooth surfaces and these thick, blocky layers, and both of those are usually remnant of ancient reef systems. There's a half-a-coral, a little two-face.
That's what it looks like when a hemicorallium gets eaten by a gonosteriid star.
Sad.
That star is munching away and has made it about halfway through that coral.
Did it kill that brittle star?
I think we're just not seeing all of the brittle star.
Because it's just like...
Yeah, maybe.
Stiff orange, not orange,
Those
yellow
stars are pretty voracious predators.
Here's would you say they're the apex predators in this area?
Outside of large fish, probably.
Yeah. There's an iridigorgia on the right. I think.
Got a nice little kind of outcrop here with a lot of different chrysogorgids, and I see another hemicorallium out there in the distance. This could be a good spot to take one of our 2 remaining Niskin bottles. Yeah, let's go ahead and trigger a Niskin here.
Roger. I'm gonna just go a wee bit ahead. Somewhere around here, all right, Meredith?
Yep, this would be great. Oh, nice. I'm sorry. Just
nice. All right, and pull right back. Roger. And then jazz hands is gonna happen. It looks like, yeah, you want 5 and 6.
Yep, 5 and 6 are all that's left.
Don't bubble on it. Better. Popped. That one was number 6 there.
Great, thanks.
And we were about 1.7 off the rock.
Perfect.
And what sample number was that?
134.
134.
Nice grab, even with schmutz in the jaw.
Looks like Hook has a little stress ball.
So as we mosey along here, off to our left and just down, there's a really large, what looks like bamboo coral.
Left and down, Raj. Good.
Bridge nav 30185.
This is a really large primnoid, probably Calyptophora.
Go ahead and start your zoom in there, please. Great.
Some squat lobsters and brittle stars.
Yep. Invertebrate associates, we call those.
Go ahead and come wide, please. Need to get myself in front.
So I remember we talked a little bit about this on one of our previous dives, but does climate change have any effect on the biogeographic boundaries for these species being observed in this location?
For species this deep, probably not yet. Deep sea stuff it takes a while for those kind of effects to penetrate down to these depths. It certainly is having an impact on biogeographic boundaries in shallower areas and on land.
There's an anemone.
Meredith, is that sort of like the gardening zones, you know, that you might see on like the back of a package of seeds at the
That's actually not a bad way to think about it.
Okay.
I mean, those are usually based on temperature and precipitation, but those are some of the same effects that'll affect what defines the biogeographic boundary. And then you can also have things like landforms, whether you're a desert habitat or a jungle habitat, and altitude all also play a role. In the deep sea, it can be defined by things like whether you're living in, say, a coastal current ecosystem, your depth strata, the average temperature in the area, and what the productivity of the surface water in the region is. And in some of these habitats, the biogeographic boundaries are actually kind of determined by what animals we're seeing. And then we're kind of trying to back calculate what the habitat conditions are that create the limitations that keep those animals in a certain region. working backwards a little bit.
Interesting. I'm reminded of, it was Alfred Russel Wallace in like Indonesia. That there's that big biogeographic line, that imaginary line that's sort of drawn in between Indonesia and Australia, maybe? Is that sounding familiar, or am I?
That one sounds less familiar to me, but I will definitely look into it. A classic boundary that happens that I am more familiar with is if you
in there, please.
are in off the coast of California, right at, and I want to say it's Point Reyes, but I always get confused, but it's right where you turn the corner to go into the Channel Islands. That's a very, very classic biogeographic boundary where a lot of the species you see north of there, you never see south of there, and vice versa. And starting there, you'll get species that extend all the way down into Baja.
Full wide, please.